Siegel. I'm the Executive Director of the Regenerative Medicine Foundation. You've heard me say that several times. I think you know who I am, but for those of you that are watching the live stream that might not be aware of that, I have to say that. I am the founder and co-chair of the World Stem Cell Summit. So, as many of you know, in this gigantic event over 4 days with some 250 speakers, getting everyone here at the right time in the right place sometimes seems a bit like an improv. But we get ourselves through this, and the lectures that we've heard this morning, I think you must agree, have been extremely valuable to understand the context of the field. I am really delighted to have on the podium now Dr. Charles Nemiroff from the University of Miami, Miller School of Medicine. And the topic of regenerative medicine and mental health is of critical importance. So many families are burdened with family members and loved ones that have depression, mood disorder, anxiety, and other issues for which the struggle is endless. The struggle seems to be endless. Our hearts go out to those families. We are so looking for solutions to move this field forward somehow, some way. And now that we are having pathfinding research using the curative, what we hope are the curative technologies of stem cells, regenerative medicine, the ability to study diseases in a lab dish, maybe we can find a way to get the right medications to the right person in a precision way. Dr. Nemiroff is a leader in the field. One of our other speakers might be joining him a little later in the session, so he promises flexibility. He has a very important lecture to give. Please welcome our friend, Dr. Charles Nemiroff.
Thank you. So I had a short drive up here from Miami this morning and braved the traffic, so I'm delighted to be here with you to try to educate you a little bit about what is truly a burgeoning area in psychiatry. In fact, we just had our national meeting in Hollywood, Florida, the American College of Neuropsychopharmacology, and we actually had a large number of sessions related to both stem cells on the one hand and inflammation on the other. So my job this morning is to educate you about a huge public health problem, a problem in medicine, and how stem cell technology can in fact be brought to bear on this. And there, obviously it's not only about helping patients, but the fact of the matter is there are commercial opportunities involved as well. So that's just the slide I use when I'm recruiting in the Northeast around this time of year. That's right near my office. I have my financial disclosures, just to be clear. I am serve on the board of a few startup companies and a number of nonprofits. All my research is supported by the NIH. With one exception, I have a stem cell grant from the Stanley Foundation that I'm going to mention to you a little later. So before I talk a little bit about inflammation, of which you know a great deal, let me talk to you some about psychiatric disorders. So the top 10 cause of death in the United States is suicide. It's the only cause of death among the top 10 that's increasing in number every year, not decreasing. Last year, in 2014, we lost 43,500 people to suicide. If you include a third of the accidental overdose deaths, which would be 25,000, then it would come out to about 68 to 70,000 a year, which we know are in fact suicides. That would make it the number 8 or 7 cause of death in the United States. So we have a huge public health problem, and much of this, as Dr. Siegel alluded to, is due to depression, post-traumatic stress disorder. We lose 3 to 4 veterans a day to suicide, so it's a big issue related to our armed forces. We lost more soldiers to suicide than combat this year. So how are we in treating depression, and what is depression? Just to give you an idea, if you think of the worst day of your life and felt that way every day and didn't know why, that's what major depression's about. It's about feeling blue, hopeless, down in the dumps, but also not being able to sleep, not being able to eat, not being able to experience pleasure of any kind, and it goes on for weeks and months. In psychiatry, we're really, really great at treating depression, I would say somewhere between 1/3 and 45% of the time. That is, we can treat patients with antidepressant medication, psychotherapy, or the combination, and they are returned to a completely pre-morbid level of functioning. The rest of the time, patients either get a little better but not well, or don't get better at all. And those people are at risk for a number of medical disorders, including heart disease, stroke, cancer, diabetes, all of which you'll recognize quickly have inflammation as a common denominator. And they're also at risk for alcohol and substance abuse. So, you know a lot about inflammation. I have little doubt about that. And you know that it is involved in a number of classic disorders, such as arthritis, inflammatory bowel disease, Crohn's, multiple sclerosis, etc. But you also know it has an intimate role in COPD, in other pulmonary disorders, as well as in Alzheimer's disease and cardiovascular disease, as well as diabetes. Now, if you look at these disorders, and you look at the basic science and the clinical and translational science, you see that there are a number of commonalities in the sense that, for example, in diabetes, there's a relationship between inflammation and insulin resistance. And as I'll tell you in a few minutes, one of the hallmarks of depression is also insulin resistance. So there have been many, many studies supported by the American Heart Association, the American Cancer Society, the American Diabetes Association, documenting the critical role of inflammation in the progression of these disorders. But what has been recognized over the past several years is that there is an intimate relationship between peripheral inflammation and the central nervous system. This is a great move away from the notion that the brain was a privileged organ that had no communication with peripheral immune function or inflammation. So we now know, and I don't have enough time to review this, but the short version of the story, and I'll show you the data, is if you have an inflammatory response, you actually experience many of the signs and symptoms of depression. Think about the last time you had the flu. You were lying there at home, you weren't working, and you had an entire day or two, but you didn't watch TV because you felt like hell, you were listless, you wondered if you were ever going to feel better again. You actually had many of the cardinal symptoms that patients with depression have every day. We also know that there is an intimate relationship between immune function and stress precipitates depression in vulnerable individuals. So for those of you who don't realize it, depression, bipolar disorder, and PTSD are largely genetically determined diseases. Of all of the complex diseases in medicine, including hypertension, diabetes as examples, psychiatric disorders are extraordinarily heavily determined as far as risk by genetics. 2/3 of the risk to develop bipolar disorder is genetic, 40% for depression, 50% for schizophrenia. And we know that there are gene-environment interactions between stress and certain SNPs and epigenetic mechanisms that control the pituitary adrenal axis and the stress response. So what can I tell you about major depression? Well, first, if you have a diagnosis of major depression, you by definition, without a doubt, will have a shortened lifespan. This is not generally appreciated by either physicians or the lay public. But the data is just absolutely ironclad. And it's not just about loss of life to suicide. It's in fact due to premature death from myocardial infarction and stroke, breast cancer, as well as a number of other factors. Women with major depression suffer from osteopenia and are at great risk for hip fracture. And the reason for that is probably both inflammatory and also due to hypersecretion of cortisol, well documented on bone density studies. So the question has been raised, is depression actually a pro-inflammatory disorder? And we've begun to learn a great deal about how the immune function, function, but in particular inflammation, actually affects the central nervous system. This may be the world's worst pointer I've ever seen. I can't even see it on my hand. So you'll have to trust me. But the fact of the matter is that cytokines in the periphery affect the brain through multiple pathways. First, there is a transporter mechanism where inflammatory cytokines can be transported into brain. Secondly, cytokines bind to receptors on the vagus nerve, and the vagus nerve have afferents that go to several different brain areas, and a number of other factors. Okay. So give me a little sign. Thank you. So this has gotten so much attention that there was a special issue of Trends in Immunology entitled Cytokines Sing the Blues, in which this literature was reviewed. So let me show you what the data are in terms of inflammation and depression. So I'll show you the first report we published, but in general, depressed patients as a group show increases in interleukin-6, tumor necrosis factor, interleukin-1, and CRP in plasma. Secondly, cerebrospinal fluid studies have shown a corresponding increase in inflammatory cytokines within cerebrospinal fluid, obviously directly affecting the brain. Now, one of the very first studies, one of the simplest studies ever done, was done by Marty Zuber, and unfortunately died of pancreatic cancer at a young age. He did a study of patients at Penn, inpatients with major depression, measured their body temperature, and remarkably was able to show that the depressed patients actually had a fever compared to matched, gender and age matched normal controls. And after they were treated for their depression, their body temperature normalized, suggesting that inflammation was a state marker of depression. So this was our very first study done several years ago, published in what's now known as JAMA Psychiatry. It used to be Archives of General Psychiatry. And you can see that we showed increases in IL-6 concentrations in many depressed patients and in many depressed patients who had comorbid cancer, but not in patients with cancer alone or in normal controls. And these findings have been confirmed in many, many studies, and I'm just showing you some of the meta-analyses. I see some of you taking pictures of my slides. If you send me an email, I'd be glad to send you the PowerPoint if you're just going to use it for your own edification or for teaching. But you can see here CRP and IL-6 are clearly elevated in depressed patients. There's a pretty large meta-analysis, as you can see, of almost 15,000 individuals for CRP and almost 4,000 for IL-6, and there was a very significant, robust relationship. We're particularly interested in this related to suicide because the field is somewhat divided as to whether suicide is part and parcel of depression or whether it has a separate underlying pathophysiology. And for that reason, suicide has been looked at quite carefully. And the data, again, in both completed suicides as well as in individuals admitted with suicide attempts, is that inflammatory cytokines are markedly elevated in these patients compared to equally depressed patients without suicide. You see this study looking at suicide attempters, and this is cerebrospinal fluid compared to plasma. Here's another study looking at blood levels of inflammatory cytokines, both IL-6 and TNF-alpha, and you see this elevation in the suicide attempters. Let me skip for time. So this is somewhat of a summary of the data that would suggest that inflammation is an important pathoetiological factor in depression in some patients with depression. So the correlation between depression symptom severity and cytokine concentration, we've begun to see differences in depression responding to antidepressants as a function of SNPs, of the cytokine polymorphisms. And then I'm going to show you some data related to what happens when you give patients, create inflammation in individuals and see what happens to their mood and how you can prevent it. And I mentioned earlier, there are a number of potential pathways through which inflammatory cytokines can enter the brain, including what are called circumventricular organs. These are organs in the brain that do not have a complete blood-brain barrier, and very well characterized. There are 7 of them. The other point I wanted to make, because I'm sure that few of you are aware of this, is that there's now a burgeoning literature that early life trauma in the form of child abuse and neglect is one of the major risk factors for depression in adulthood, as well as PTSD, and moreover, that child abuse and neglect is associated with an increase in inflammation, which lasts for the lifetime of the individual and likely is one of the reasons why adults exposed to severe neglect or abuse as children have a shortened lifespan due to risk for medical disorders. So this is a study that we did a few years ago, taking men who had been abused as children and who were now depressed, and we looked at the IL-6 response to a very mild social stressor. And you can see here in red this profound increase in interleukin-6 concentrations in the men who were depressed with a history of abuse compared to the normal controls. So there is a signature, a memory that results in a hyperactive inflammatory response in depressed patients with a history of early life trauma. This work was confirmed in a series of studies done in London at the Institute of Psychiatry, the Maudsley, using a cohort of individuals followed in New Zealand. And the data is pretty striking. If you look at the upper left corner, those are CRP data, and it shows that CRP clearly remains elevated in individuals with a history of early life trauma and probably underlies in part the risk for cardiovascular disease. So the question is, do pro-inflammatory cytokines contribute to the syndrome of major depression? And is that something that occurs in patients who only have comorbid medical disorders, which are common, like cardiovascular disease or cancer, or is it true in major depression per se? What are the mechanisms, and most importantly, what are the treatment implications? So we used interferon alpha as a provocative way to produce a robust inflammatory response. This is a treatment for patients with malignant melanoma, and it used to be a treatment for patients with hepatitis C before the new treatments were available. So we know that if you give interferon alpha to patients, they're going to have a huge inflammatory response in terms of interleukin-6, TNF-alpha, and other cytokines. So we took these patients and we treated, these were patients with malignant melanoma who were getting interferon alpha, and you can see that they had very high rates after treatment of depression, anhedonia, the inability to experience pleasure, sleep disruption, all of the difficulty concentrating, all of the cardinal symptoms of depression. And we decided to see if we could prevent the depression by pre-treating them with antidepressants. So we pre-treated them beginning 2 weeks before the very first infusion of interferon alpha. And this was a 12-week study which we ended up publishing in the New England Journal of Medicine. So this is what depression looks like in the patients who received interferon alpha and placebo. And you can see that 60% of the patients ended up getting depressed. That's syndromally depressed by 12 weeks. Many were actually suicidal. But if we pre-treated them and kept them on antidepressants, we could protect all but 20% of the patients. So it looks like this is a reasonable model. And we went on to do a number of studies, I'm not going to bore you with the details, showing how increases in inflammation mechanistically produce depression. One mechanism is by reducing the availability of tryptophan, which is the rate-limiting step in the synthesis of serotonin, one of the major neurotransmitters involved in depression. Another is by further accelerating the major mammalian stress response to the pituitary adrenal axis. And we were able to show that in non-human primates, interferon alpha produced a depressive-like syndrome, just as we saw in patients. And then we were able to go on and do brain imaging studies and show that increases in inflammation are associated with changes in brain areas that subserve both mood regulation and psychomotor function, and correlates pretty significantly with fatigue, as you can see here. So the good news is that this treatment is now reserved for patients with malignant disease that have not responded to other chemotherapy agents. It's no longer used to treat hepatitis C. So if depression is uniformly elevated in patients with autoimmune and immune disease, and if depression is a risk factor for disorders that we know involve inflammation, then we really need to ask ourselves whether the subset of patients with depression could be treated with a potent anti-inflammatory agent. So, let me see if I could. The first study ever done was done in a group of patients with comorbid depression and psoriasis, and it used the TNF-alpha antagonist, one of the inflammatory cytokines. And very oddly in this slide, higher numbers are better. So these were patients who had failed antibiotics. They were randomized to a TNF-alpha antagonist or placebo. And in fact, even if you didn't respond in terms of your psoriasis getting better, the TNF-alpha antagonist had antidepressant properties, so administered intravenously. That led to a second trial that one of my students was in charge of. This was a study with Infliximab. Infliximab is a TNF-alpha antagonist, and I want to show you the interesting results of that study, and then I'm going to swing back to our stem cell study that we're just initiating. So TNF-alpha antagonists have some antidepressant properties in preclinical models. This is a monoclonal antibody that has to be given intravenously. And so when I was in Atlanta, my colleague Andy Miller took the lead in this study, and we wanted to look at patients who had failed antidepressant treatment. So there were 60 patients, 30 were randomized to placebo and 30 to infliximab. They received an infusion at baseline, as you can see at week 2, and another at week 6. And this was almost a classic antidepressant study in that you can see the age of the patients. They had no comorbid medical disorders. they were on no other antidepressants. They were not, of course, suicidal. Could never get them into a study. Their mean age was in their early 40s. In almost all depression studies, 2/3 of the patients are female and 1/3 male, because depression, the epidemiology is exactly that. Women have depression twice as often as men, and you can see that 40% were unemployed. And they had suffered quite some time. Their mean age was 42. Their age of onset of their depression was in their late teens, early 20s, as you can see, and they had suffered multiple episodes of depression over the years. And the results at first glance were extraordinarily disappointing. This shows you the antidepressant response over a 12-week period, and it looks like there's no effect at all. And when Andy called me to tell me the results, he was virtually in tears. And I said, well, what about their baseline inflammatory markers? So we haven't looked at that yet. I said, well, do it. And here's what we found, was that if you look at the bottom left-hand corner, the patients with the highest levels of inflammation, namely CRP levels above 5, had a great antidepressant response to Infliximab, and the same with the patients with the CRP level above 3. So that if you define a subtype of depression with depression plus inflammation, and then use this TNF-alpha antagonist, you may in fact have a decent antidepressant for that patient. And I'm just editing a new journal called Personalized Medicine and Psychiatry for Elsevier, and this is exactly the kind of data that we're looking for. So why am I here? What does this have to do with your life in stem cells? So Josh Hare is the director of our Stem Cell Institute, and unlike many cardiologists, he's attuned to the fact that many of his patients have comorbid depression. And so he's seduced me into the Stem Cell Institute, and I've become somewhat of the psychiatrist, one of the few psychiatrists in academics that has an appreciation of and a desire to study inflammations and the potential role of stem cells as therapy. So, you know, I've read, you know, all the papers you all have probably read. I've probably read hundreds of papers about stem cells in regenerative medicine and their remarkable anti-inflammatory effects and how they quiet down inflammation by 15 different mechanisms, including reducing pro-inflammatory cytokines, increasing the expression and production of anti-inflammatory cytokines. And so, you know, I was involved with Josh, and I forced Josh to measure a crude measure of well-being in the patients he treated, both in this MI study, but also in his frailty study. And in both studies, mesenchymal stem cell therapy was associated with an improvement in well-being, very crude measures. So we have now proposed to do the first study of mesenchymal stem cell therapy for the treatment of refractory depression. What we're going to do, unlike Andy Miller's study, is we're going to pre-select patients who have elevated baseline levels of inflammatory cytokines, who have failed previous antidepressant treatments. We're going to randomize them to mesenchymal stem cells or the placebo infusion. We already have pilot data from Josh that we could reduce TNF-alpha and CRP levels for a minimum of 6 months after a single infusion of stem cells. So the Stanley Foundation is a mental health foundation founded by a family that wants to develop novel treatments for psychiatric disorders, and they've just awarded us a small grant, about $750,000 grant, to do a pilot study, and I have a grant submitted to NIH currently to look at patients with comorbid depression and alcoholism. And the reason I did is because alcoholism is associated with flagrant robust inflammation. Depression's associated with the same, and it's the kiss of death together, and patients with depression and alcohol abuse do not respond very well to our current treatments. So this is the design we're going to use. It's very simple. It's an 80-patient study. We're going to admit patients who have failed at least 2 antidepressant treatments, and then we're going to randomize them to mesenchymal stem cells or placebo. We're going to follow them for a year after the initial phase of treatment. Because it's a biologic, we don't know what the time course of response might be, but we're quite excited about it. So I think the data together has shown us something pretty interesting. First, I think there is an optimal amount of, the inflammatory response is not there evolutionary for no reason. Inflammation is not always a bad thing, right? And so it didn't surprise me that in the patients that we gave a TNF-alpha antagonist to who had normal baseline cytokines, they actually didn't do well when they were given the TNF-alpha antagonist. And it suggests that there's a sweet spot for inflammation vis-a-vis mood. And I think that's what we have to go for. There's also, you should know, an IL-6 antagonist study that J&J is currently conducting. So let me summarize, and we can get ready for the next speaker. There's no question that a sizable percent of patients with major depression do have a very clear and robust increase in pro-inflammatory cytokines. There's little doubt about that. In our hands, this is not first episode garden variety major depression. These are largely patients who have failed antidepressant treatment and have been chronically depressed and/or have been victims of child abuse and neglect. We know that stress, which precipitates depression, also precipitates the inflammatory response. There's clear evidence that inflammation is depressogenic, and anti-inflammatory response apparently is associated with an antidepressant response. So I think we may have to add one more category to our classic views about inflammation, because depression seems to be part and parcel of that syndrome. And I'm going to show you the sunset of Key Biscayne. Thank you very much.
I'm going to call up our next speaker, and if there is a little time for a couple of questions afterwards, after our next speaker, I appreciate you staying here, Dr. Nemiroff. Thank you. And this session is to end at 10:30. So I want to introduce our next speaker on this important topic. Dr. Steve Walker is an associate professor of genomics and molecular biology, systems biology, at the Institute for Regenerative Medicine at Wake Forest School of Medicine, led ably by our friend, Dr. Anthony Atala. So, Steve, thank you for joining the program and adding to this important topic.
All right, well, thank you for the invitation. It's a pleasure to be here. This was kind of last minute, so please excuse me if the talk looks a little bit last minute put together. I'm going to talk about a project that a graduate student of mine had been working on. She's now matriculated, so I'm hoping to find someone else to pick the project up. But we're trying to develop a model where we can look at the enteric nervous system using iPSCs, patient-derived iPSCs from patients who have an enteric nervous system dysfunction. So my lab focuses on, one of the projects we focus on is looking at autism spectrum disorder children who have a GI disorder. And so as you probably know, ASD is defined as a neuropsychiatric condition, so it's defined by the DSM-5 rather than, there aren't molecular and biochemical markers to date. But there are a whole lot of inputs that result in autism, and so we're trying to pick some of these apart at the moment. As you probably are aware, autism is associated with a number of comorbid medical conditions. One of the primary questions is it a brain disorder, or is it that affects the body, or is it a whole body disorder that impacts the brain? And so one of the areas we're focused on, autistic children experience GI symptoms at a higher incidence, prevalence, than their neurotypical peers. And this happens in autistic children at a relatively early age. So the question is that impacting neurodevelopment? Or that's the question we're looking to answer. So what we know is there are a host of studies that look at GI disorders in autistic children, and they report the prevalence to range from about 20% or no different from neurotypical peers all the way up to 70%. So that would be an extremely high number. 70% of autistic children have GI dysfunction that's debilitating, and it impacts them at a much earlier age than their neurotypical peers. Then it's plausible that that may impact neurodevelopment. And it's been reported now in numerous publications that there's a positive correlation between the severity of autistic symptoms and the occurrence of GI problems. And another interesting facet is if autistic children with GI symptoms are treated effectively for their GI symptoms, often the clinician will note an improvement in cognition and/or behavior. Sometimes dramatic, sometimes not so dramatic, but the bottom line is these GI problems are not your typical upset stomach. These are dramatic. You have teenage autistic kids still in diapers and that sort of thing. So it's a chronic, terrible condition for the family, the caregivers, and the child, of course. So if you can fix that problem, even if you can't do anything more about the autism itself, you've eliminated a huge burden on everybody involved. So that's the approach we're taking, is to look at the GI symptoms. Number one, see what we can do. I guess we had to make a case in the medical community and in the scientific literature that GI symptoms actually are severe in autistic kids. And one of the problems is kids going to a pediatrician, autistic kids especially, are largely or often nonverbal, and they have unusual or odd behaviors to start with. And so if you go to the pediatrician and you, as a parent, and you explain your child's doing this, that, or the other, often it was just dismissed as autistic-like behavior. And there are specific things, if you're a gastroenterologist, that you can note about these children, even nonverbal children, in their behaviors that suggest an underlying comorbid medical condition. And in fact, there was a consensus report put out in 2010 through a group at Harvard and in collaboration with a whole lot of other folks who perused the entire literature on GI symptoms in autistic kids. And one of the findings was that the single piece of information that you may have about an underlying GI condition in an autistic child is unusual behavior, which seems kind of counterintuitive because they have lots of unusual behaviors. But if you know what to look for and you find it, and then you treat the underlying GI symptomology, then oftentimes you get an improvement in behavior and function. So are GI disorders and neurodevelopment linked? Well, again, one of the interesting things, and it's not widely accepted, but certainly the clinician I work with who treats exclusively autistic kids now, we have a tissue bank that includes samples from about 1,500 children that he's seen. The average age of the children in his practice are 5 or 6 years old. By contrast, when we went to get control samples for the molecular studies that I'm doing, the average age of those children going into a typical pediatric GI practice are about puberty age, so 12, 13 and up. You don't see a lot of 3, 4, 5-year-olds in a pediatric GI clinic, at least not at our place. And so the opposite is true in the autism clinic. You see the majority of kids are less than 10 years old. So that's striking. And in fact, some kids are as young as 1 and 2-year-old having chronic GI symptoms. And by chronic, we mean bad enough that you need to go and see a doctor about them. And So it's tempting to wonder and suggest, if you have a chronic GI condition at 2 or 3 or 1 year old, is that impacting neurodevelopment? And in fact, does that precede the onset of autism or vice versa? And so that's an interesting possibility. So are they linked? Well, obviously there's a gut-brain axis, and the gut and the brain communicate. And so that's a place to look. There's been a leaky gut hypothesis around for a long time. So it's been known, not just in autistic enterocolitis or bowel disease, whatever you want to call it. IBD in general often is manifest as a leaky gut. So things that are not meant to get out into the bloodstream from the GI tract do. And if you imagine a child having that condition and stuff getting out of the gut into the bloodstream and then making it perhaps to the blood-brain barrier, and in fact maybe even breaching the blood-brain barrier, then it's not too much of a stretch to think maybe there might be a relationship between, because at that time, of course, it's very active neurodevelopment and 2, 3, 4 years old. So it's an interesting hypothesis. It hasn't been proven one way or the other. I mean, you can demonstrate that you have a leaky gut, but you can't necessarily yet conclude that that causes brain dysfunction. And the other thing, again, there's a temporal relationship. So the onset of GI symptoms in this population often happens much earlier than in a typically developing population. So again, it's tempting to speculate that maybe that impacts neurodevelopment. And so how do you test this mechanistically? Well, this was the question that we had. So when I got into the field of autism, I'm not a neurobiologist, I'm a molecular biologist and geneticist by training. So all the literature 10 or 15 years ago started out by saying autism is a brain disorder, and it's a lifelong hardwired brain disorder, and that's where you start. And so I thought, well, there's nothing I can do in this field to impact. I mean, autism is extremely interesting because the very cool thing is, if as researchers we can do something to reverse or slow down or make it so children never get the diagnosis, then you impact the entire lifespan. And that's pretty exciting if we can find out what it is why these kids are particularly vulnerable to this neurodevelopmental problem. So one of the first meetings I went to, they talked about a number of topics related to autism, and somebody gave a talk about GI disorders in autism. So this was 15 years ago. And that really struck me because, again, this person also did a small study in a small cohort, but found there was an extreme overrepresentation of GI symptoms in the autism cohort. And they were very young. And so this person speculated that there might be an impact on neurodevelopment. So I thought, well, that's an interesting place to look. And then fate would have it, I hooked up with this GI doc who was very interested in treating ASD children who had GI problems, and so we've been doing that ever since. In the meantime, so I'm talking to my graduate student, how do we test whether or not the GI symptoms impact neurodevelopment? Well, there is a condition called Phelan-McDermid syndrome, genetic condition. So by definition, PMS or PMDS patients are all lacking a part of the long arm of chromosome 22 that contains a postsynaptic density protein called SHANK3. So by definition, they're all lacking some part of that, and in some cases there are other accessory proteins on the long arm that are also deleted, but at a minimum they are haploinsufficient for SHANK3. And so these children who have Phelan-McDermid syndrome experience developmental delay, intellectual impairment. And again, these are sort of heterogeneous. I mean, they all experience some level, some more severe than others, and it may depend on how much of the chromosome 22 is missing. But the bottom line is they all have intellectual impairment, absent speech, hypotonia, GERD, gastroesophageal reflux, and then cyclical vomiting. Okay, so the interesting thing is we looked at PMDS patients. 84% also have a diagnosis of autism spectrum disorder. And greater than 40%, maybe 50% also have a GI condition, GERD. So now we have a genetic condition that's ripe for model building, I think. So we kind of thought this was stepped right into our lab. So what does SHANK3 do? Again, at the bottom part, it's a postsynaptic density scaffolding protein. And if you're haploinsufficient, it's already been demonstrated to be involved in a number of neuropsychiatric conditions, including ASD. So the goal of our project, and again, this was my graduate student who was developing this model, the PMDS patients diagnosed with ASD and GERD, we were going to use those patients. So here's the point. SHANK3 has been studied extensively in the brain, in autistic patients and also in the PMDS patients. And it's known to cause hypoactivity at the synapses. And so there is a connection, there is a research connection to what a SHANK3 mutation or haploinsufficiency is doing in the brain that may impact neurodevelopment. We're interested in the enteric nervous system or the gut. A lot of the ASD kids, whether or not they have Phelan-McDermid syndrome, experience hypomotility. So in the enteric nervous system, SHANK3 also acts in the enteric nerves, but most studies to date have looked at the central nervous system. So the novelty of this is we're now going to look at SHANK3 haploinsufficiency in the enteric nervous system, and we'll do that using cells derived from patients with Phelan-McDermid syndrome, so we know they're haploinsufficient for SHANK. So in a nutshell, that's it. So we think the lower esophageal sphincter, so below the esophagus, the constructs of these PMDS patients will have less, so we have hypothesized because they're haploinsufficient, the ENS neurons will also have less SHANK3, and so we'll just measure it by molecular means, and then we'll do physiologic measures to see if in fact this hypomotility is related to, and we think it certainly must be related to the SHANK3 haploinsufficiency. So there are just 3 goals of a project, and I'll go through these rather quickly, but the first goal was to derive iPSCs from patient, and this is more and more common in ASD and neurodevelopmental disorders and all sorts of disorders. You take, excuse me, cells from patients who have the condition, you make induced pluripotent stem cells, and then you can take them down neural lineages or any lineage that you're interested in studying. And they already have the genetic problem built in, so now you can study, hopefully, physiology and molecular biology. So the second goal of the thing is we'll differentiate these patient-derived cells into neural crest cells, and then the final step is we'll put those neural crest cells together with a muscle construct. They'll innervate the muscle, and then we'll measure physiologically whether or not the synapses are working properly. So derivation of these iPS cells, so there is an NIH stem cell repository, believe it or not, for the PMDS population. It's a relatively small population, but just like autism 15 years ago, these foundations and stuff spring up because parents have children that are affected, and they start foundations so that research will get done, and they're very motivated and very active. And so in the autism field, this has now resulted in Autism Speaks, one of the largest funding agencies in the world for autism research. The PMDS, it's a much smaller population, but no less impacted, and so the parents have now formed a foundation, so the Phelan-McDermid Foundation, and they have donated cells from their children to the NIH repository so that we can do studies like this. And so again, we picked, not all the PMDS patients have autism and or GERD, so we specifically picked PMDS patients that had both for this study. And they were male, because again, autism affects males at a ratio of about 4 to 1 compared to females. And so what they have at this repository, and also for the autism studies, there are a couple repositories that take beta blood cells, and they immortalize the blood cells to make what's called lymphoblastoid cell lines. And the reason for that is then they can freeze those cells away and then grow them in perpetuity, so you can use them indefinitely for studies and continue to divide and ship them out. So we use these lymphoblastoid cell lines. There's some limitations with using them, but again, the genetics of the cells should mimic exactly the donor that they came from. So we have some experience with this. We made lymphoblastoid cell lines. When you make iPSCs, and this is relatively new, there are a couple papers out that have done it, and we're actually in the process of publishing a paper where we specifically use autism-derived lymphoblastoid cell lines and made iPSCs. When you make the iPSCs, you need to then get rid of obviously the viral factors. And so there are techniques and ways to do that, and so that's where we are. And it's just a standard protocol. We've been able to make iPSCs from a couple of the PMDS cell lines that we got. And we, let's see, yeah. And we evaluate the cells now using a new PCR-based assay that allows you to measure pluripotency factors and then factors for each of the germ cell lineages. And you can tell at what stage, whether your iPSCs are truly pluripotent still or whether they're starting to differentiate. And so we've actually incorporated that, and we're actually publishing a study just using this assay rather than the teratoma assay that is standard. The second goal is then differentiate these into neural crest cells, and then we'll characterize. And so this is the stage that we're at. This hasn't been done before, as far as we know, to make neural crest cells from iPSCs. And so it's a little bit tricky, and the project sort of stopped when my grad student left, so we're going to try to pick this back up. But that's the goal, is to make neural crest cells. We can make the neural rosettes, but making the neural crest cells is a little bit trickier. And then the third goal would be to mature and differentiate these. And there is evidence that this has been done already. We have a chap at the Institute who developed a model system where you take from the mouse on the right, you take muscle cells, and you can culture the muscle cells, and then we take the neural cells from the Phelan McDermid patient, and we co-culture those so that we have the stem cells, we have the muscle cells. They end up looking like this, and the nerve cells eventually innervate the muscle, and then you can measure physiologic criteria on these constructs and characterize them in all the standard way, and we can then look and see if these patients or these constructs represent a haploinsufficiency of Shang3, how much is there, where is it, what is it doing, how is it functionally acting, and so on. Sorry. This is from her thing. Okay, so again, our goal is to develop a model to look at enteric nervous system function as a proxy for CNS function in a Shang3 haploinsufficient population, and sort of to ask the question: are the two related? And more importantly, does this impact neurodevelopment? And is there anything we can do to correct that at an early time? Okay, that's it. Thank you.
We have time for a couple of questions.
Yes, thank you very much for the excellent discussion. I have a question for both speakers. Could you comment on the use of mesenchymal stem cell therapies for autism particularly, knowing obviously that we have trials in IBD? Thank you.
So it's no secret that parents with autistic children are very prone to a variety of alternative treatments. And therefore, both in Mexico and in certain centers in South America, parents have brought their children for treatment. The data is sparse, to say the least. And the data of inflammation, putting aside my colleague's presentation about this variant and whether inflammation may in fact mediate the GI symptoms, the evidence that autism is primarily or even in part an inflammatory disease is not compelling. So the idea that one could give stem cells that in some way would act as if this was a neurodegenerative disorder and that you'd be replacing neurons that were somehow lost in development. The neuroanatomical studies of autism have remarkably not shown very many differences in terms of volumetric studies of brain regions. So it's not like a neurodegenerative disease where you could look and say, oh, the hippocampus is reduced in size and we need to find a way to increase hippocampal neurogenesis. So what I tell families is that the data simply isn't there at this time, and I certainly can't control what's going on outside the United States.
And I would agree with that. And in fact, you know, a lot of the autism meetings, there are, depending on which meeting we're talking about, there are people that are representing the stem cell side of this. And I have to agree, there isn't any compelling even rationale for why what stem cells might be doing to ameliorate symptoms or conditions associated with autism, with the exception of the inflammatory GI condition. And the reason I say that, I met an MSc expert a couple years ago, came to our place, Arnie Kaplan, and he talked about a study that they did using his company, his former company, I guess, Osiris. They had MSCs, and they did an MSC therapy for IBD. And if I understand him, if I remember right, he said a single bolus of MSCs in Crohn's patients had dramatic, remarkable impact or effects. Now, I don't know what the follow-up is, but when I mentioned to him this IBD-like condition in kids, in these kids, and it looks a lot like Crohn's in some ways, it also looks like ulcerative colitis, I thought, wouldn't it be interesting? So that's one way I think maybe stem cell therapy might be, you might be able to apply it to that population, but that would be to impact the GI condition, not the brain condition.
Any other question? One more question, yes.
Question for Professor Nemeroff, please. So basically, I really enjoyed your presentation because of the correlation between the depression and the inflammation process. And as I understood correctly, depression might start at young age or whenever later, but at young age with abuse and everything, and then it would lead to inflammation. Whereas later on in life, it could be the other way around. Inflammation can lead to depression. And as a patient who received the stem cell shot, I did feel like a major euphoric side effect. But the question is to you. Young people, lots of times, are even more depressed because of the cause and effect surroundings. They don't know what's going to happen in their life and everything, and they're maybe more suicidal than middle-aged people. Why don't they? Why are they more depressed if they have their own stem cells in the body because they're still growing?
Right. So remember, well, first, there's a bidirectional relationship between inflammation and depression. So increases in inflammation will induce depressive symptoms, and depression is associated with, in fact, an increase in inflammation. So it's bidirectional. The issue that relates to age of onset has to probably do with genetic risk. So there's no single gene for depression, but there are about 15 SNPs that have been identified, some of which actually include inflammatory cytokine SNPs. So I believe that most of the patients that we see with early onset depression, putting aside bipolar disorder, which is largely in the teens and early 20s, are people who probably just have a genetically bad hand. They've had some early life traumatic experiences and more recent life stressors. And that's the perfect storm in terms of inflammation.
I want to thank my speakers on this very important session. Let's please give them a round of applause. We're going to have in this room an announcement of our poster award winners in the next couple of minutes. All of you eager young people that are in the room and postdocs and